We have our exam a week from this Thursday. So next Monday and Tuesday, USM has off for fall break. So we have 2 class meetings before the exam. We'll talk about that today. We're in good shape. And Muhammad just asked about the idea of that old first exam. So, and I talked about it in the recording I made for class last Thursday that I missed. And we'll talk about all that in a second. But just to answer Muhammad's class question while we're recording this, yeah, so I shared in announcement number 6 on Brightspace the first exam from last year. And it's important to look at that to expect to see what to learn for this exam. I don't have an answer key because I found that if I provide an answer key, students just like, yeah, okay, I'm good, I know that. You're not going to be able to answer all the questions because last year's course was slightly different. There's a little bit different emphasis, so so it's a thing. But if you have questions about it, let me know, and we'll go through it because it's very important to see that. So this is where we're at, and we're going to talk today. We'll talk about Thursday, and we're both adding new material this week and reviewing as you get ready for for the exam because we don't have a class a week from today. We have spring break, right? So we'll have today and Thursday, and next time I see you, a week from Thursday, it'll be exam time, okay? Just before I forget, if you haven't watched the answer key video for quiz 2, you should watch it because I do coaching when I do these answer keys. And one of the coaching things is, on quiz 2, students were writing long answers, like repeating the question and writing long answers. And the exam is designed for you to be able to get it done during class time, but you gotta move through it. And you gotta be aware, this is a 2-point question, so I'm gonna give it a 2-point good answer and move on like that. So again, that quiz 2 answer key video, I say you gotta watch that no matter how well you did on quiz 2. And it's there, and watch that because there's that. Now, then the other thing that Muhammad just asked is, am I going to provide a, what did you call it, like a study guide for the exam? And the answer is, and it sounds like a smarty pants answer, and I apologize, but everything I say in class is the study guide. So some students have come up after class and had their textbook, and they're like, wow, the textbook has way more stuff than we talk about in class. And so I've described this already, that the class time I have is just so limited that everything I say is super important. That's a big reason why I record it and share it, so you can get it, even if you have to watch it again. Like that atavism, right? When we talked about that dolphin that had the back flippers, people were like, whoa, well, what's an atavism, right? And it's an evolutionary throwback to an ancestor. The dolphins don't look like that anymore, but they don't because they have some interesting ancestry that had limbs, and dolphins evolved a initial embryonic development of a little hind limb bud that then regresses, disappears because of programmed cell death. And sometimes there's a failure and they get a little hind limb. Very rare. But it's just a key thing. And how many points is that? What's up? Fill in the blank, right? What's an evolutionary throwback? Atavism, like that, right? And so this is a study guide, and I know that's you're like, oh, come on, like that. But I just couldn't shrink it down, like that. And so, so that's the thing of that. It's just you really have to get stuff that's going on in class. And I say the purpose of our textbook is that find the stuff we talk about in class in the book, and get that so it helps you understand what was expected in class. You get some kind of confirmation from an official textbook. Yeah, this is important stuff. Do that first. Don't just start read. You don't just study by reading your textbook, page 1, page 2, page 3, page 4. And if you get really solid on the course class meeting stuff and how it is helped in the book, then read the book. And that's great. Because biology is always more stuff. It's just more stuff. And so the challenge is to try to learn more stuff. Now, I'm going to ask a couple of small point questions on the exams. So the last thing to study is that extra stuff and navigate through that. And don't think I'm going to ask some trick question. I don't have any trick questions or like a little footnote, a little detail. It'll be like, oh, here's a box, a page big box in the book. We didn't say anything about it in class. Well, what's the key thing there? Like that, right? And so that's the answer to that. Thank you, Muhammad. And so, sorry I missed class. I was sick, and I made a recording on Friday. So I was still sick on Friday, but I'm recording because we just have to keep going forward. It's really important for you to watch that. If you haven't watched it yet, don't just like, oh, I'll watch that when I'm studying for the exam, because class meetings always have it in sequence, you know? So I'm like, okay, we're moving on. I talked about stuff on Thursday. I also gave assignment that's due this week on Thursday. So what should you do this week, okay? So this is the last piece of what we're gonna cover, and we'll do it on Thursday for the exam. And it's little parts of chapter 5. Does anybody need to read all of chapter 5? No. Just these little parts. And so it's, I don't know, 7 pages of chapter 5, and then, 5 or 6 figures. And that's what we need to get out of chapter 5. So our exam, we know what it's covering now, everything that's going to be on the exam. Okay? Like that. And we'll talk about it, but write down questions. And then, I haven't gotten too many of these chicken embryo model building assignments. I've gotten several, but I haven't gotten them from everybody. And they were due last Thursday while I was sick, and maybe some people thought, oh, I guess that assignment is just gone with the wind, But you gotta send that to me. And we're not physically handing them, and to take pics of your labeled chicken model assignment like that. Because then on that Friday's missed class meeting recording, I say, oh, and then we're having an assignment for this one. And it's a drawing assignment, and it's a big drawing assignment that's in chapter 4 about this Wnt signaling pathway. And it's a really important concept for us, and so we want to get that done by this Thursday. And I put markers outside my class. I shifted it around. There's lots and lots of colorful markers in there to make a big drawing. So you're welcome to come over there and do it. If you have art supplies at home, great. The key thing here is to make a big drawing. So that's what you're going to recall. And so like tape 4 pieces of paper together to make your drawing, and just make it big. It's really obvious that students, most people, have like a hesitation to draw big because they're not artists. And this is not art. This is just a simple drawing. But make it big, use different colors, because that's what your memory will be when you recall it. You'll bring that up, and if you spread it out, that's great. You can just have simple labels on it for the assignment. But I mentioned before, you could make it big enough that you actually have a flashcard that you set down, and the flashcard is Wnt. And what's Wnt? And you flip it over, and it says Wnt is a signaling factor. There are orthologs in every animal on our planet. Wnt is way more significant in our lives than our heart, or our blood, or insulin, or hemoglobin. Because if we didn't have Wnt, we would not have been born. It would just not have gone even a little bit down the pathway of forming an embryo. So it's a super important one, and it's an example that we'll return to in the same way we turn to, and we say, let's talk about myogenin, because you guys know there's myogenin gene, and it can get activated. It can get transcribed. How does it get activated? It's in every cell. How does it get activated? And the answer is, this MyoD transcription factor protein binds to it and activates it. That's a universal example of a gene. Wnt is a universal example of a signaling factor. So we want to be solid on that signaling pathway. Question of the exam could be just draw it and label. So the exam will be like, repeat these things, And so in the recorded lecture that I made Friday for a class meeting that I missed, I say, let's just look at that exam 1, it's an outcome number 6, and I just show a couple of examples. Because what you're expected to do on this exam is nothing like multiple choice, nothing like, oh, I see that, that's it. You actually have to recall it and describe it. Again, briefly, directly, as best as you can. So think of the assignments. And right now, what could you say about the vertebrate phylotypic stage? What are the 10 hallmark features that we learned, and what are each of those about? And that could easily be a 20-point question on our exam, where it's 10 points for the names of them and another 10 points for just brief, what is that thing, What's the notochord do? Why is that listed? Yep. And you can do that, or it might be kind of an image of that, the figure that you use to build your chicken model with little arrows and fill in the blanks like that. And then it's just huge. It's a drawing of your body a long time ago. You can point to so many of your body parts right now, right? Heart, bone, like that. Well, this is you a long time ago, and those parts were super significant, and this is, kind of probably the first time you get to learn about that. And it's not just our body, it's great. We're focused on our body. A lot of students want to go into healthcare and so forth for careers, but also it's every vertebrate that's alive right now. Go on a nature walk and see some fish migrating, going like, well, they had a phylotypic stage, they look like ours. Some differences. Here's something I want to just pick up on because it was in the news in the world yesterday, and you're like, whoa, and that happens every time I teach this course because this is a modern field of science, and it's just roaring forward. So stuff comes up where you're like, whoops, have to just insert this in our course. So, and this is also a good point of, hey, I say, remember, we focus very early on a couple of little packages of knowledge about this nematode. So, right, you're going to that thinking, right? When we learned about programmable cell death, and you learned about there's some orthologs, and we learned about caspase 9 and B cell 2, right? And so recall those. You can probably pull up some images. We learned some terms of genetic analysis. We had that assignment of looking at a genetic screen. I can have questions on the exam where I ask, what's a loss of a functional allele and so forth like that. And I like it that my mind goes to this nematode worm. We have this programmed cell death in our body. It's really important in our health and disease and cancer biology and so forth. But I always start out thinking about what I know about the nematode and programmed cell death, and I've just added everything I know about cell death to that place. And so that's that. Then remember we talked a little bit about this RNA interference, okay? And so it's that second story, and this is this is fascinating to me. So in 2006, so almost 20 years ago, those two scientists won the Nobel Prize for discovering that RNA interference, okay? Because it was just it's a big deal. They had done the work basically 10 years before that. In the 1990s was when scientists really discovered this RNA interference, the microRNA, the siRNA, and these guys won Nobel Prize. And I was alive then, and my friend Ann Rugby's like, well, why didn't her boss win the Nobel Prize? Her boss was this guy, Victor Ambros, and he studied this before those guys did. And he studied it in C. elegans, like that. So I lived through that, and it was really surprising. That's why this guy's hitting his head, because he's like, what the H? Why am I picked? Because he wasn't even the superstar, like that. But we don't have to worry about that. We don't have to worry their names. It's just kind of good to know that it was, not that long ago in human history that this was discovered, okay? So let me just show you this, because for a long time, I would just go, RNA interference. And slowly over time, in the last 20 years, it's kind of the field has gotten shaken out as it gets bigger. It's a natural process. It happens in us. It happens in every animal. It happens in every plant, so on and so forth like that. So it's a natural way of regulating gene expression. It's always a repression of gene expression, a decrease in gene expression. And it includes both that natural process and then the scientists realizing they could take advantage of that. They could design experiments in a lab, and they could knock down gene expression for any gene they wanted to study, okay? So there's really kind of two parts of it that are cool. It's important, it's normal, and then you could take advantage of it and do stuff in experiments, right? And so over time, people have started to distinguish more and more between the siRNA and the miRNA, okay? And so I just mentioned that briefly. I didn't used to talk about it, but people are paying more attention to that. So when we say siRNA, there's a couple of features of it that are details that are shown up here. For us, siRNA really targets RNA degradation. So there's a target RNA that's going to get repressed because it's going to get destroyed. Then there are these microRNAs, miRNAs, and they tend to not destroy the RNA. They bind to the target RNA and they block it from getting translated into protein. The effect is the same. The gene is silenced. It gets transcribed, the RNA's there, It might get degraded, it might get blocked, but it's a gene repression mechanism, okay? It's just so cool. So that's what these guys actually won the Nobel Prize on in 2006. And at that time, nobody, even the winners, really were distinguishing between these two stories. Like that, you know? They're just like, yeah, great. You know, they won in microRNA, you know? My friend Ann, her boss studied that. And she's like, why didn't my boss win the, you know, like the super experts, the winners? They were like, well, I don't know what's going on, okay?
What does the SI stand for?
Small interfering. Yeah, and you don't need to know that. And this is like a 1-point question at most. It's to distinguish it. It's just a little nugget that goes in there. But again, it's world news. And you're like, what the, huh? And yeah, yesterday you're like, wait, yeah. So yesterday I was like, what? Yeah, okay. So then the next thing to say about them is that actually both occur in nature. The SIR story is less common in our bodies than the miRNA story. So it occurs in nature in all plants and animals. It's just in general that SI degradation of the RNA story, that's less common, less examples of that in nature, and many more examples of this microRNA and just blocking translation. When scientists take advantage of this RNA interference in a lab, they always do this siRNA strategy because it's really easy to measure levels of RNA. And if the RNA gets degraded, you're like, yep, we knocked it out. That's what they want to confirm, right? They build a little RNA interference, and they're going to try to knock down expression. And they can confirm that by just measuring the target mRNA levels, and they get destroyed. They disappear. This one is just harder to study because it's the protein that doesn't get made, and it just takes more work to study the proteins. So scientists always do that. And so yesterday, the Nobel Prize in Medicine and Physiology was awarded to Ann's boss and Gary Rupkin for working on, and see, they say microRNA. So it's just crazy because, again, the earlier people actually got it for, now we see it's siRNA and the degradation. And they got it 20 years ago, 18 years ago, because of its value in experiments. That was the first thing that Nobel Prize people said. It's like, whoa, that's nuts, because you're going to be able to use it in lab and really go to town. And that's happened. And now, yesterday, these other scientists got it for the more common natural mechanism. Because over the years, people have studied that, and they're like, oh my gosh, there's so much of that in nature. For us, it's just a detail. Again, I only recently started distinguishing between small interfering siRNAs and microRNAs, miRNAs, in classes because it's kind of a detail for college. But then it just jumps out in world news yesterday. And people are just shocked because they thought, well, you know, Ann's boss didn't get a Nobel Prize. And then he got one. And he's just a goofball. All these people are just goofballs because they all studied that nematode worm, which is just like not a winning thing. You know, it's just like if you told your grandma that's what you're working on, you know, at Thanksgiving, she'd just be like, okay, you know, loss of, you know, you were really good at school, like that kind of thing, you know, just like, oh, I thought maybe you were going to be a doctor, and you're like, well, kind of a doctor, I'm a worm doctor, Grandma. Yeah, so it's just fabulous, right? And in that announcement, there's just, in the news now, there's just stuff in the regular newspaper that's just straight out of our class. Like, look at this. Every somatic cell in the body has the same set of genes. Yet a brain cell is different from a liver cell. Differences in gene regulation across development drive cell diversity and the emergence of specialized cellular functions. And you're just like, whoosh, right? Because that's just right out of our class, right? So it shows, again, that all this stuff is super important. And we're just scrambling to introduce it and help build a framework. The book has too much details. If you read the book, you would just start forgetting stuff in class. It's too much, you know? So you really want to get this framework and try to hang on to it because people are talking about it today. People are wandering around. Ann Rugby lives in St. Paul, Minnesota.
Did you message her?
She's partying. I didn't yet, just because Ann is partying. You know, she's out just partying and dancing in St. Paul. And yeah, okay. So just loop back on that, just for not because we needed to, but just because it's just striking to see that, the immediacy of the stuff that's happening in our course. For us, here's that thing for the first, again, I turn it sideways because it's a little bit of a timeline, and it's just universal gene expression in our cells and eukaryotic cells. Here's the gene, it's always there. Here are the transcription factors. They activate, we get transcription. That's the first step in gene expression. Then, this is where those RNA interference steps occur. And some genes have them, some really important genes, but all genes have to get this. And so our course really focuses on this. MyoD is a big time transcription factor. We'll talk about this a bunch. In development, transcription regulation is the first, it's always the first step. In development, it's a really common step of regulation. It's like, we're just not gonna make those genes until we need them, right? So, you know, we got the genes to make muscles and we're just not transcribing those genes until it's time and transcription factors get activated and do that. Now, to catch us up to where we're at in our course now is especially focused on the idea that our first example of MyoD is MyoD gets made and it immediately starts to work, do its job. One thing that happens a lot in development is that the gene gets expressed, but it might not do its job right away. And so it's going to get activated. And an example I used in the lecture on Friday recording is the estrogen receptor and the testosterone receptor in our bodies. We have both, all of us do, and all vertebrates have both the estrogen and testosterone receptor. Those transcription factors are present in our cells our whole lives, but they are turned on by a hormone, and the hormone doesn't occur in our bodies to significant levels until puberty. So that's an example of a transcription factor that gets made, ready to do its job, and then it pauses at that step for years when we're growing as children, and then we reach puberty, the hormone shows up, the transcription factor does its job. And that's really common in development, to create that kind of timing, right? The genes don't just get turned on all at once, boom, 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 like that. There's delays and steps. And so that's really common. And what we're transitioning to is that lots of times it's a signal from another cell, like a hormone, that activates expression in a target cell or population of cells. And that's what that Wnt signaling pathway is about. That one cell or a group of cells makes this Wnt, it releases it, and some target cells have a receptor. The Wnt binds, and what does the Wnt do? And the answer is it activates a transcription factor. And the transcription factor is really the famous thing. It's called beta-catenin. It'll be in your drawings. And it turns on genes. In the case of beta-catenin, what happens to it is that normally, in the absence of Wnt signaling, that protein just gets broken down. It just gets degraded. So it's just in our cells. It's always getting made, but it's always getting degraded. And Wnt signaling stabilizes beta-catenin. So it's just really, it boils down to, we're just taking our clay model videos and we're saying, let's kind of make those fancy. So instead of just having the transcription factor like MyoD and then myogenin immediately do their job, let's build in a delay. And the delay is very often a cell communication. At the start of our course, I said that our cells got to become different from each other, and there are just two ways. One is unequal cell division. We've seen a couple of examples of that. Stem cells, and also that mitotic cleavage in early frogs, where the cells are not dividing equally. And now we're going to have a bunch of examples of the second way cells become different. They become different by cell communication, cell signaling. And so Wnt is an example of these cells are going to start becoming different. And we'll learn some of the names of the signaling molecules. In other cases, we'll just appreciate they're becoming different because of signaling. For example, we said there's the notochord, and the notochord is famous, and it induces the central nervous system. And it does that by releasing some signaling factors. And the signaling factors activate transcription factors in those ectodermal cells, and the cells change from preparing to be skin cells to folding up into the neural tube and becoming our central nervous system. So we might sometimes name the molecules. Other times we'll just be like, yeah, that's cell signaling happening. That's the story. And what we're doing now is, in chapter 4 and 5, the book drops down to that molecular level and wants to talk about some of those signaling molecules to introduce that. And it's done that, so again, we're just building up on that. And it's done that in one case, in the first chapter, it describes this experiment, and it's an experiment where there must be some signals that were released. So we have the cells transplanted 100 years ago by Hilda Mangold between these two salamander embryos really early, and some of the cells could create this conjoined twin, an identical twin on the tummy of the host. And they do that by releasing signaling factors. And that's this dorsal lip of the blastopore. That's going to be important, and it's important because it's releasing signaling factors, and that's activating gene expression. So it activates gene expression in the tummy, and it changes the fate of the tummy cells. The tummy cells were going to be pretty boring, just skin, and then on the inside, there's that somatopleura, this mesoderm that's on the inner lining of our coelomic cavity, our gut cavity. So, you could take a knife and flap your body open like this. And the flaps have the ectodermal epidermis on the outside, pretty boring, and the somatopleura mesoderm on the inside, pretty boring. When we transplant the cells from the dorsal lip of the blastopore to that area, those transplanted cells release signaling factors. We don't have to know the names of those, but we do need to understand that what they do is they change the gene expression in the host ectoderm and mesoderm. Those cells can change, and they change from going to becoming a fate to be like just boring skin to the brain and spine. Or on the inside, they change from just being boring inner lining of the gut cavity like that to things like making the heart and muscles like that of the body, bones. Yep.
Have they tried this with other animals, or is it just the salamander?
So that's a good question. And remember how we said that if you, in the first clay model videos, we said we could make a modified transgene and put it in. We could force expression of the MyoD in like a precursor to cartilage, and it would make muscle. So the answer is yeah, it's
Gotcha.
been done a bunch of times, and it could be done kind of in the olden days by a transplant. And in modern days, the scientists will just manipulate gene expression. And it's all based on the reality that, these cells, they have all the genes, and we can turn on some of those genes, either through normal development or through a transplant, where we add some different signals like that. Yep. Okay? And so goes back to the very start, and we keep looping back to the very start of our course, because there are just a couple more things in chapter 1 that we just really didn't nail yet, because the author, again, just wanders through the whole parking lot and points out lots of really important stuff. So I made this, I just photoshopped it, so it looks like it's a figure of your book, and it's not. And it just shows the reality for every animal and every plant. It's a multicellular plant or animal that started out with a fertilized cell, and then these are mitotic divisions, and they're going to make the organism. And there are only two things happening, growth and patterning. And our course really focuses on this patterning, and patterning looks like that. Importantly, all those cells, they have all the genes. So patterning involves changing gene expression, and it often involves changing gene expression by having a cell signal like Wnt regulate a transcription factor like beta-catenin and change what genes get expressed. and that happens naturally, and you can mimic it in a transplant experiment, or you can mimic it in an experiment where you manipulate gene expression. And that's all happening in that Hilda Mangold experiment. She's doing a salamander, but it's the same as a frog. And so we actually spent a lot of time, and you're on this early development. There's the phylotypic stage, and we talked about cleavage and forming the blastula, then gastrulation, then some about neurulation. And so there could be a figure on the exam where it's like this, or it could just be an open-ended thing, say describe in words, like a short essay, frog development that you could see under a microscope. So you don't have to mention any genes, just what you could see under a microscope from fertilization till the phylotypic stage. And I'd be looking for blastula, mitotic cleavage, gastrulation, blastopore, blah, blah, blah. You know, like that? Deuterostome, yeah. And I'll get different answers, you know, so I don't have just a rigid rubric in these, because I'm looking for 10 points, and I'm like, whoa, those are 10 good points. You know, you're good. And then somebody else might have an overlapping set of answers. That's absolutely fine. That's my job to roll through it. I'll make an answer key video where I talk about that. But yeah, just be like, yeah, I'm learning some stuff, and I'm going to be able to say what I learned, you know, on the exam. And someone will be like that. What do you remember about that? So you can practice that, you know, just look back. Because some of the stuff we've learned about is big and chunky, like the 10 different features of the phylotypic stage and what they do, or early frog development, or we're learning now at the molecular level. And so in the Friday video, I start talking about this handout, and we want to get to that and talk about that today. And you'll just want to memorize the handout. The handout is what Mohammed asked about. It's a study guide. A study guide to like a 20-point question about describe frog molecular development, starting at fertilization, and describe, not this words, but just molecular details. Okay, so we'll get to that. And where we're at is, I started talking about the handout a little bit on Friday, so make sure you catch up with that if you haven't. And we're going to talk about this, and we're going to get it to there. So that's what the handout is. And it's just about frogs, so the story is somewhat different than in our body. Right? We don't have that early development the same. Some of the proteins that we'll talk about are orthologs. Beta-catenin comes up. Like that, okay? And so let's get to that. And these are just some figures from chapter 1 and 4, where the book is introducing this idea of, here's the target cell, and this is the extracellular environment, and here's a signaling molecule, like a hormone, or like this Wnt, or other signaling factors. And when it's present, it finds a receptor that activates a pathway that really often regulates a transcription factor. In this case, the transcription factor is inactive until it gets phosphorylated. It gets a phosphate added to it. There's lots of different ways of regulating proteins, switching them on and off. And we'll learn a few of those. For us, the key thing is the idea of the signaling molecule that's going between cells and the changing gene expression and the transcription factors that regulate it. This in between is less important, but we start adding some to it because, of course, that's the pathway that has to happen inside the target cell. The book in chapter 1 shows this. It introduces different types of signaling between cells. That's another figure from the book that's probably still in chapter 1, okay? And so I also described this on Friday, that when we see this, this is a fate map of an early blastula. So just to show you that, what we're looking at is this time. So it's after fertilization, and it's this early blastula here. And we can tag these cells and say, what are you going to become? If we just leave the embryo alone, what are those cells going to become? And the answer is, well, we want to pay attention to the ectoderm, mesoderm, and endoderm. And this is a fate map. And so we talk a lot about this idea that the early frog embryo is a sphere, and it has like the globe. The North Pole is the animal pole, the South Pole is the vegetal pole, and the equator is this mesoderm. So you see it's a belt of mesoderm, and that's got to fold in. And that's a big focus of the handout, because this is what the plan is in the early blastula. But you can do experiments also to say, do the cells know what they're supposed to do? So one experiment you can do is in the early blastula is you can just take cells out and have them by themselves in a dish and say, do they become mesoderm? Even though that's their fate, is that what they know that's supposed to be? And I talk about that in the recording because I made this. So this isn't in your book. This is actually a really good thing to see. This is when you do the experiment and you say, take it out of the early embryo. Even though these cells are fated to become a belt of mesoderm in the early blastula, they don't know that yet. If you take them out of the embryo, they don't become that. They just become boring ectoderm. What do we know? They must receive a signal. So the handout is all about that. What's changing in that belt of cells, and how is it changing? And the answer is two things. When you see that belt, I'm going to start talking about the belt buckle. That's going to be the dorsal lip of the blastopore. That's a really special place where those cells, their job is going to be to release these signaling molecules. That's what could change when we transplant the cells to the tummy of a host. So we got a belt of mesoderm and then a special buckle. And so we have really two stories. And one story is, let's make a belt. And you can kind of see the hint of it here. There are going to be molecules here that mom puts in the egg when mom's got just the egg in her body. And those are going to basically float up, like from the South Pole, and they're going to signal these cells to become mesoderm. And then that's a story like that signaling pathway we're talking about now. They're somehow going to have a signaling molecule bind to receptors and change gene expression, so these cells start to become mesoderm. Then these buccal cells, they get those signals too. They're like, yep, it's supposed to be mesoderm. And then there's a story in them where they're special mesoderm. And that's the first part of the handout. In the first part of the handout, soon after fertilization, even before the first cell division, a protein gets pulled up to the buccal region, the eventual buccal region, called disheveled. And disheveled is going to stabilize beta-catenin. So that transcription factor we're learning about in the Wnt signaling factor story plays a role here. And it plays a role to make the special buccal. Okay? And so let's look at this on the handout. There's words and then just this one image from a different book. And the image has 3 numbers that are numbered on the handout. So you're going to be sort of 3 steps. And the first step we're going to talk about is to make the buccal, this region here, we're going to stabilize that beta-catenin. Normally it just gets destroyed, and we're going to stabilize it. And then beta-catenin is a transcription factor. So it's going to bind to a gene, a bunch of genes, but we'll learn one, the Siamose gene, and it's going to activate its expression. And interestingly, before beta-catenin does its job, there's already a transcription factor on the beta-catenin regulatory region called TCF3. And TCF3 is repressing that Siamose gene. The Siamose gene is in every cell, and it's off. Every cell, and it's off. And it's being kept off. You only want to make a buccal in one place. And this is the buccal maker story. So let's keep it off. And so beta-catenin is an interesting transcription factor because what it does is, it always does this, it overcomes repression. And we get a real molecular switch. Remember I said this course is like car mechanics? And on the first night of the class, the car mechanics instructor takes us to the parking lot, and we want to study cars, that's like the human body, but the instructor has like lawnmowers there. and other kinds of simple motors. Because he says, look at this example. And he flips over the mower, and you're like, oh, wow, I see the mower cuts grass by spinning a blade around an axle or a pivot. And he says, that idea of things turning around an axle is really important. The car has that in many, many places. So that's what's happening here. The beta-catenin orthologs are present in every animal on our planet. And it always is the same story. It's a transcription factor that creates a really nice molecular, a really pretty molecular switch, because the target genes for beta-catenin are off. They're being repressed by TCF3, and beta-catenin switches them on. So it's not just kind of a lazy, you know, like, oh, kind of like a light dimmer. It's a light switch. They are off, and now they're on. And just to show you that, so Siamose gets made, and Siamose, the story is like that myo-D, myogenin. Siamose is a transcription factor that immediately does its job. It's only made in the eventual buccal region. There are Siamose genes everywhere, and they're repressed. So we only see the Siamose RNA and protein in that region that's going to become the buccal. And this is going to be turning it into a buccal. The Siamose is a transcription factor. The protein gets made, and it immediately activates another target gene, glucocorticoid. And I know these are really dumb, weird names like that. They just are. You know, the people who named them are just goofy goofballs of the world's greatest degree. And for us, for now, we just want to see that the activation of glucocorticoid involves this pathway, like dominoes falling, and it just gets a little bit complicated because there's another story that's happening in the regulatory region of glucocorticoid at the same time. So we have another way of creating a molecular switch. And what this is like is this pathway is essentially, say, via buccal. This pathway we're going to talk about is going to be coming up from that bottom part, from the south pole, and say B mesoderm. So we have this kind of combination double whammy effect on glucocorticoid, where glucocorticoid gets activated saying both B mesoderm and be the special buccal of the mesoderm. Okay, so that's a visual of what we're going for here. The big picture is, yep.
What is the disheveled protein?
Disheveled?
Yeah.
Yep. What is disheveled? Yeah, so that's a good question. So in the Wnt drawing assignment, the assignment is to show how Wnt can activate beta-catenin. And in the assignment, the way it activates beta-catenin is it activates a protein called disheveled, and disheveled protects beta-catenin from degradation. So that's just a little bit of this idea that we're especially interested in a signaling factor like Wnt and a transcription like beta-catenin. And once in a while, we'll mention some of these steps. And disheveled is one of these players on this step, because it's going to stabilize beta-catenin. That's how we get beta-catenin active. Okay? So let's look at this. So this is the handout. I just put all the words on here, and you have the handout. And and this is just where that figure is, but I just left the figure off. So the handout is these words. And so, and I talked about this first part on that lecture on Friday. That was from Miss Class. So I was going to talk about this and explain that. So we're just going to jump down here, and this part is the part where it's coming up from the south pole. So this first thing describes how we're going to get beta-catenin activated, and the second part, and that's going to be the buccal, and the second part that we'll talk about now is how we get the cells in this region to say we should be mesoderm. And it's going to be signaling molecules that are going to be floating up from that south pole, that vegetal pole. So let me just show you this image again to remind you that the cells early, they're fated to make a belt, but they need to be in the embryo to do that. If we take them out of the embryo early, they don't know that they're supposed to be a belt. They've got the genes to be a belt, but they don't know. None of the cells know they're supposed to be a belt. They need to receive some signals. They need to be in the embryo during that blastula time to get these signals, and we'll talk about them. And in talking about them, the book talks about these two, and the book talks about all this in a more detailed and kind of chaotic way in chapter 4. And so I've tried to give us something that we can really, sort of hang our hat on and learn. So let's just talk about this, and we'll talk about this first one, and then we'll have a little concept check. So it says, and talk about down in this area, it says veg-T, it's a protein, it's a transcription factor. It's a transcription factor that is a maternal factor. And I defined that in that recording on Friday. Maternal factors are factors that are loaded into the egg by mom, while mom's making the egg. And in these non-mammalian vertebrates and invertebrates, they have big eggs. And we know in a chicken egg that you eat at lunch, there's a lot of nutrients. And we know mom loads in a lot of nutrients, a lot of building blocks. Well, in those big eggs, mom is also loading in a lot of these regulatory factors, and veg-T is one of those. Veg-T's got a job to do. And it's a maternal factor. That means it was created mom before mom met dad, right? The eggs were formed, and these factors are located. So these are what factors that are maternal factors. It's initially found as mRNA in the vegetable pulp. So let's just start out, and I was doing this in the recording on Friday. Oh, and here's still some of that images. I was saying, here's an egg, a mature egg, before it's fertilized. There's a little sperm coming to it. And I talked about disheveled in that recording, and I talked about GSK3 and how GSK3 is responsible for degrading beta-catenin, and beta-catenin gets degraded everywhere in the early embryo, except for that part that's going to become the belt buckle. So now we have another one, and so let's just add that here, and it's in the mature egg. So before the fertilization, that we have this veg-T, veg-T. And it says in the egg, mom actually loads it in as mRNA. So there's veg-T mRNA. It's a maternal factor. It's loaded as mRNA. And that's just a little example to tell you that when mom makes the mature egg, she makes everything ready and waiting. Not going. We have to put a big pause because we don't know when mom is going to meet dad and fertilization will happen. So that's true for these big eggs, is that they have these regulatory factors and they've all paused and they're waiting for fertilization, and then they go again. And this one waits because mom doesn't make the protein, she just makes the mRNA. And there's no translation happening in the big mature egg. It's waiting for fertilization, and then the ribosomes activate, and they translate all these RNAs. So this is a way of making it pause, and it's a cute way of making it pause. Veg-T protein is not going to do its job because it's not even made yet. And after fertilization, it gets made. So if we did this, what we'd see is that after fertilization, so now we have this early embryo, now the mRNA starts getting translated, and we get veg-T protein here. And it says it's a transcription factor, so we know what it wants to do. It's going to bind some target genes and activate them. What kind of target genes is it going to activate? It's going to activate target genes that are going to make signaling molecules that are released and go up and tell these cells to be belt cells. So it's a really sweet story because it's not a lot of pieces. And then another thing we remember is that during early development, the frog embryo is going through this mitotic cleavage, this unequal cell division, this mosaic development, and these cells become special because these cells are the only cells that have veg-T protein. These are the only cells that activate the signaling molecules that are going to, say, be a belt. Yep.
So is this process, I assume it's very similar in humans, but we're just
Yeah.
using this now?
Totally different. Yeah, and that's one thing that you should just be out, and I told you before, when you see a mammal, like a squirrel, just go up and pet it, don't worry about getting bit really hard. You know? I can tell you that in North Yarmouth, the sheriff shot a rabid fox off his leg this summer, because I know the people who lived at the house. And the fox was on his leg. They were chatting with the sheriff. They're like, a rabid fox around here, and it's under the deck. And then they're just chatting because the fox is under his deck. And the fox comes out, and it's rabid. And I've never seen a rabid animal, but if you do, it's pretty wild. Because the fox just charges the sheriff. And the sheriff's going like that, and shoots the fox off his leg, right? So I don't know why I said that, but anyway, don't mess with that. Oh yeah, pet squirrels. Because mammals are totally interesting, because we have tiny eggs with apparently none of these maternal factors. Mammals make these early embryos that are all embryonic stem cells. They're all equivalent. Such a different thing than all of our non-mammalian vertebrate relatives, like frogs, right? Those were our evolutionary precursors, and becoming a mammal was really a wild thing, because of how much the early embryonic development changed as part of saying, oh, you don't need to have all those nutrients, you don't even need to have those maternal factors. Okay, so let us pause for a concept chat and see where we're at on that, and we're doing great. Yeah, rabid fox, talk about that in your group. Rabid fox shot off his leg, you know? Like, that's a good shot. I don't even know if he drew his pistol. He might have just had it in his holster and shot the fox.
I never got, I was scared.
Oh, okay. I think I posted it on the web. I'll post it again. So Alex says that, they didn't get the handout. I will post it. I think I posted it before, but I'll post it again.
Okay.
Yep. You guys had to make 2 groups of 2. Mohammed and Jeff, you guys had to be 2 groups of 2. Yeah, just so, because it's better, because you're like, okay, good. Yep, don't worry about it. Yep.
I mean, I like saw how it looked like. I'm gonna get my No, I did. I know that's something that
We'll just do another minute.
I think it's measurements, right? Is it just 2 Oh, I think it's smaller.
And let's see, Alex, what is one thing that your group talked about?
Talked about how the siRNA causes degradation and the miRNA causes inhibition of protein synthesis, and how the siRNA degradation is less common in nature, but used in experiments.
Awesome. And so that popped up just a little bit in our class earlier on our course. I just mentioned it, and when I asked about it in the test, maybe 1 point like that. But then it popped up in the news yesterday, so I wanted to loop back on that just because it's just a wacky story, but it still isn't a huge thing for our course, right? When we talk about RNA interference, it's part of it. And clearly, you know, the world thinks it's a significant part because we got 2 sets of Nobel Prizes. I mean, that never happens where they're like, oh, we got to go back and give some Nobel Prizes to Ann's boss. Then Alex also said that they didn't get the handout, so I handed it out one day, and I will post it. I think I posted it that day, but I'll post it again today, the handout. And I don't have it physically with me. I have more copies of it. I'll have those on Thursday. I actually left my textbook at home, so epic failure on my part. And then, Jesse, what's one thing your group talked about?
We were talking about the veg-T mRNA and how essentially it's just crucial for the development of an embryo and what.
Yep, that's awesome. Yeah. And so with that veg-T, when you see that, right, in that early frog embryo during that mitotic cleavage, we're just like, we have that big egg, it's fertilized, it's a single cell, now it's going to be 2 cells, 4 cells, 6 cells, 8 cells, like that, you know? And as we're doing that, the cells are not going to be equivalent because there are a variety of these maternal factors, like veg-T, that are in different locations. It's kind of like you imagine a pie, and you're like, we're going to make a blueberry apple pie, but we're not going to stir the apples and blueberries. And when you slice it up, you're going to have different slices of pie. That's what the mature egg is like. Yeah, it's got a lot of nutrients, a lot of building blocks, you know, amino acids, blah, blah, blah. But it has these maternal factors that are not equally distributed. And so we go a long way toward starting to build this early frog body just by the way mom builds the egg in her body before she ovulates it, before she releases it from the ovaries and releases it in the environment for the male to, fertilize. Yep, yeah. And so they're just saying they're saying it's such an important concept of building a mature egg and having it wait until fertilization, and then having fertilization kickstart all these processes, these many, many processes. And one famous one is veg-T gets loaded in just as the mRNA. Something that's fascinating about that is there are a bunch of different mRNAs that are loaded in, and they're not getting translated, and there's a reason why, and it's a detail. mRNAs get a long tail added called a poly A tail, and in the mature egg, the poly A tails are short, and you need a long poly A tail to get translated. So fertilization causes polyadenylation of all these. And it's just a little taste of that because students immediately are like, Zach, and go, wow, is this like on our body? And we're like, whoa, no. You know, it's just fascinating to see the difference between a mammal and a frog, of course. But one of them is the start of embryonic development is totally different deal, you know, like that. So do we have an ortholog of veg-T? And I'm like, probably not. If we do, it's doing something totally different in our body. You know, it's like if it's doing it, it's like a wrench, and it's not doing that wrench job. It's doing some other wrench job. Yeah, like that. Yep. Other questions people have?
I do have one.
Yep.
So we've learned about the animal and vegetal regions and how they're kind of different, especially in the frog. Are they different? I know that this is very different from humans. Are the 2 regions distinct in the human development too, or no?
No. So another thing you mentioned, I said you can get identical twins in mammals, but not in anything else. Because when a frog is just the egg, it's already uneven. You know, it's like that pie, and you can't slice the pie in a way to get 2 blueberry apple pies, because the apples didn't get stirred together with the blueberries, and we have them in different places. So you just can't make identical twins. If you did that with a frog, you'd get sort of like the rear end of a frog and the head of a frog, and you'd be like, ooh, you know, sort of like that. You get 2 different parts of the body. Yeah, it's really interesting. Okay? Other questions people have? And so again, you know, it's this kind of nuts and bolts, kind of like a car mechanic story where I've just tried to, you know, make this really crisp. And this is why veg-T? Well, 1, it's a transcription factor, so it's going to activate genes down in that region. Then 2, it's ready to go. It's a maternal factor, but it's not really ready to go. It's just RNA. And then it gets translated, and we get the transcription factor going. So it says veg-T is a transcription factor, it's a maternal factor. This he found as mRNA in the vegetal pole. Veg T activates transcription in vegetable cells of genes related to nodal that are signaling factors. So down there, after fertilization, the Veg T protein gets made, and then it gets to do its job. And its job is to bind to the regulatory region of genes called nodal related, and those genes are going to get secreted. Those are going to get made into proteins, and the proteins released from those cells. And that's an example of cells that are the source of one of these patterning signaling factors. And they just float out from the South Pole, and they reach this region, and they tell those cells to be mesoderm. Okay? So these cells also secrete VEG1. So just another example, because it turns out our book goes through many, and we've just boiled it down to as minimal as we can to get that core framework, and maybe there'll be a point on the exam to add on it. But if you can repeat something like this on the exam, I'll just be like giving you a high five. I'll find you on campus and be like, rock it. Because these vegetable cells also secrete VEG1, so another signaling molecule that comes up from there. And these signaling factors activate other transcription factors not shown in the figure above. So let's just go to that figure one time. Oops, not that way. And show you this. I said that when we activate this glucocorticoid, it's the buccal activation story, the beta-catenin story that's told in number 1 on the handout. And then it also is these other guys coming up to say, be mesoderm, be a belt. And that's this pathway coming in. And what we talked about is these cells receive those signals, and they're like, we should be mesoderm. We're going to change gene expression and be mesoderm. And mesoderm, yes, and special mesoderm belt buccal. And it's just a great analogy because it really is a belt, and it has a very special part that's like got that extra function of a belt where you're like, yeah, you have to have a buckle. Okay? So that's what this is driving at. And it's just saying that, several signals come up to make that happen. They all kind of team up. And then so it says VEG1, and it says 3. This VEG1. It turns out that's a famous signaling molecule because it's in a gene family, and it's in a gigantic gene family that's called TGF-beta. And in our bodies, we have orthologs of that family. So not necessarily, exact like VEG1, we don't have that same function, but we have that family. And in us, there are like 40 members of that gene family. So that's our second signaling factor. We're learning about Wnt. In our body, I think that gene family has 16 members, numbered, because it's just really useful. It's like a screwdriver, and you're like, we could use some more screwdrivers with slightly different shapes. We can get a lot more done, build a fancier body. And this TGF-beta family is the same thing, 40 members, just number. They have some different names too, like that, but numbers. And so just to underscore that, it's a TGF-beta signaling factor that is loaded as an inactive pre-protein in the vegetable pole. So let's go back here. We're doing great. We have a few more minutes.
When it says loaded, does that mean it's like a maternal factor?
Yep. So it's made by mom, and she puts it in the mature egg while she's getting ready to go on a date. Sorry. But true. And so let's go to this. And so again, I don't have space. So I'll erase this, but it's that same idea of let's go to the mature egg and see what mom is getting set up here. And because some of these proteins are maternal factors. So we draw this, and then there's the sperm, so not yet fertilized. And this is that VEG1. And here's VEG1, and it says it's a protein. So there it is. It's a protein in the mature egg. So it's a maternal factor. And if you haven't watched that recording from last Friday, maternal factor actually has a really interesting definition. It's not that mom made it. The definition is dad can't help. So if mom has a mutation in a maternal factor gene like VEG1, the embryo is going to be defective because dad's genes have not showed up yet. And that's really what a maternal factor is. A maternal factor is a genetic term, not a development term or biochemistry term. Maternal factor is a gene that can't be rescued by dad's genes. So even if dad has wild type VEG1, if mom is mutant for VEG1, the embryo will be mutant, because only mom matters. So that's a good, and again, you know, I don't want to loop back on Friday and just repeat it, because you got it. And you know, if you haven't watched it, try to watch it by Thursday, like that, okay? So there's VEG1, and in this case it says, we want to tell the story a little bit too here, because mom also makes these maternal factors that are actually made as protein, so there's the protein, but it actually is made as an extra long protein that keeps it inactive, and this is called a pre-protein. And the pre-protein is the original full length of the VEG1 protein.
What does that say up at the top?
Mature egg.
Oh, okay. Thank you.
And so there's the protein, and what needs to happen after fertilization, this pre-protein and a variety of other pre-protein maternal factors gets clipped. It just gets cut here, and that little extra piece floats away, and now the VEG1 is now the functional protein. And its function is just to be released from these cells. They just let it go, and it floats up and helps tell these cells to become mesodermal belt cells. So that's why, you know, why do we talk about VEG-T and VEG1? Well, just because there's some different stories there that tell us just a little taste of all the preparation that mom does. In human history, kind of the famous situation is, remember, the first people that looked under microscopes at sperm literally drew a little person driving it, because that's the understanding, you know, just 200 years ago, is the sperm matters. The sperm is active, it's wiggling around, that's obviously where the little person is, and the egg looks like it's just food. But the opposite is true. The sperm is showing up and kind of coming to the party late with just some DNA that some of them, you know, like, oh, can't help with VEG1, sorry, you know, I love you, but I'm sorry, you know, like that. And mom's just set it all up, and it's all ready to roll like that, you know? So we got to do a little bit of that, right? Because we're talking about these early embryos. And let's see, we have 5 more minutes. So that's just that. 3 is just that. Just another one of those that's going to be coming up from the vaginal pole to pattern that mesodermal belt, and just add some little knowledge about that. That there are a bunch of these maternal factors that are loaded, that are made by mom and put into the maturing egg in her ovary that are pre-proteins. And fertilization gets cooked, and they get to do their job. Okay? So then the 4th one. And this one goes back to the drawing. So just glance at the drawing, and it goes back to just explaining, now back to goosecoid. And again, the first number on the handout tells about the cynomolus transcription factor binding there, that purple protein binding there. And then 2 and 3 told about the signaling pathways. And the signaling pathways are going to activate that transcription factor, and we're not learning the name of that transcription factor, we're just learning that these cells must respond to the signals and activate that transcription factor, and those guys have to team up. So we get a nice switch. It's like you should be a belt buckle, you know, where you need to be belt, mesoderm, and be that special buckle.
Is it not just VEG1 and cymose?
So is it not just VEG1 and cynomolus?
For the transcription at the
No.
stage 4, okay.
So that's a good question, because what we're just not learning here is that these signaling molecules, so nodal related and VEG1, they're coming up from here, and what they do is they bind to these cells, and that activates transcription factors inside the cells.
Okay, gotcha.
So it goes back to that drawing of the signaling molecules typically just come to the surface of the cell, bind a receptor, and then there's some activation of a transcription factor inside. And just in our last couple of minutes, let's just clarify that so everybody has a good handle on that. So here's a cell, and here's the nucleus. So it's in a eukaryotic cell. And we started our course, we learned about transcription factors. And so transcription factors, they do their jobs in the nucleus, because their job is to bind to DNA. And transcription factors are not outside the cell. They're not going between cells. They're in this cell, and they're doing their job in that cell. Then, really pretty purple signaling factors, signaling molecules, they're outside the cell, and they typically do not come into the cell. So these are, and developmental biologists just call these generally signaling factors. And so what we want to emphasize in our course is there's a way for these signaling factors to affect those transcription factors. The signaling factors come between the cells, like they come up, they're released from the vaginal pole, they come up and they bind to the belt cells. They don't go inside, they bind to a receptor on the cell surface, and then there's a signaling pathway that affects the transcription factors. And so it's a little bit confusing to start with, because they're both called factors, and that's just such a generic word. But there's the visual, that transcription factors are in the cell, they're in the nucleus, they're doing their job, that's what their job is. Signaling factors are outside of cells, going in between, floating between cells. And so that's why we have that Wnt drawing assignment, because the Wnt drawing assignment is this. You're going to put labels on your Wnt drawing assignment, but that's what you're really trying to show is, Wnt is coming between the cells, and it's going to regulate beta-catenin, which is going to be stabilized and be in the nucleus and do its job. So that answers your question, which is those signaling factors, nodal related, and VEG1, they float up, but they are these purple guys that float up and they reach the belt region, but they don't go into those cells. They bind to receptors and the belt activates those transcription factors that are ones we're not going to name, because
Gotcha.
We've got a good bunch of stuff we're learning. So here's the pathway. See, it's a signaling pathway. It's going to activate that kind of blue transcription factor, and it's got a name, but we could name everything, and then you guys would just be in the corner throwing up. Yeah. How about that though? A sheriff just shot the fox off his leg. Well, he's chatting with these. It's in the middle of the summer. He's just chatting with these people on the deck because they're like, yeah, it's pretty nice out, blah blah blah. And they've got this rabid fox onto the deck, and eventually the rabid fox just comes shooting out and just climbs the sheriff's leg, and the sheriff goes, choo. And then they keep talking, because we live in Maine. They just keep